The topic of discussion today is spinal poisons. Now, first of all, we need to know some basic physiology to understand this type of poison. See, this is a motor neuron. All this is happening at the level of spinal cord. Now, this motor neuron controls this skeleton muscle contraction. Now, it also gives a signal to this Renso cell, which is an inhibitory cell. Now, this Renso cell will release glycine, and this glycine will act as an inhibitory neuron on this neuron. Why? Because if this neuron was not there, then this would have caused excessive muscle contraction. So to prevent this excessive muscle contraction, this motor neuron gives a signal to an inhibitory neuron to act as an inhibition on itself. Now, in the strychnine poisoning, what happens is that this strychnine, it blocks the glycine receptors, so there is no glycine acting on this neuron, and there will be excessive muscle contraction. In the tetanus poisoning, the tetanus, what does it do is it inhibits the release of glycine. So both are doing the same thing. Glycine is not acting on this neuron, and there is excessive muscle contraction. This strychnine is a spinal poison, and the tetanus, it is not a poison, it is a disease. Now, what is tetanus? Basically, tetanus is, let's say you are hurt, or let's say there is any injury due to a metal, and it's deep inside the skin. So in that, the Clostridium tetani bacteria enters the skin. That bacteria release a toxin called tetanoplasmin. The toxin travels retrograde manner to the spinal cord. So injury happened over here, and the injury is like deep inside the layer of the skin, so the bacteria can invade, and the toxin travels retrograde manner in the nerves, and through the nerves it reaches the spinal cord. And at the level of spinal cord, what happens, I told you that it inhibits the release of glycine. So basically, tetanus is also doing the same thing. And how strychnine reaches the spinal cord? Strychnine is the poison. We ingest the poison, it goes in the stomach and then gets absorbed in the blood vessel. Through the blood, it comes in the spinal cord. Through the blood circulation, entire poison is at the level of spinal cord. So both are ultimately reaching at the level of spinal cord. Both are doing the same thing, stopping this inhibition over here, right? So now we'll study. Okay. So now what is this? Because the muscle inhibition, because the neuronal inhibition is stopped, there is excessive muscle contraction, which results in this posture. You can see there is hyper extension of this spine, and it cannot relax, it is contracted in that state only. This is what, this is opisthotonus. Second one is recus sardonicus. Here you can see that these facial muscles are contracted in that state, they cannot relax. This one is recus sardonicus. Now what do you think, the person is conscious or unconscious in this? The contractions are happening, but they are happening at the level of spinal cord. Nothing is reaching the brain, so no consciousness is altered. Person is conscious during these contractions. So let's start about our spinal poisons. The first one is, the plant name is Strychnos nux vomica. Strychnos nux vomica. The pronunciation is a little bit difficult. We'll try it again. Strychnos nux vomica. And in this plant, the active substance that I told you is what? Strychnine. Yes. So this is also known as Cuchilla plant. Now it has a fruit, orange color fruit, inside which there are seeds, 4 to 5 seeds. Now the intact seeds are non-toxic, and the seeds are called Cuchilla seeds, dog's button or Quaker button. But if you ingest crushed seed, then even one is fatal. But intact seeds are non-toxic. Active principle, as I told you, the active principle here is strychnine. I explained what strychnine does at the level of spinal cord, right? Other active principles are brucine and logenian. So now strychnine mechanism of action. I already explained you this, that it blocks glycine receptors. Then there is no Renso cell inhibition and uninhibited muscle contraction. There is high muscle contraction which is causing those postures, rectus sardonicus and the facial features, because the muscles are contracted in that state. Now, as we all know, strychnine and tetanus poisoning, they resemble each other because the place at they act are the same. And the third one is oxalic acid poisoning. We are not studying anything about it, just we have to know that it resembles them. Now, now, as I told you that one question is the posture opisthotonus. Is it seen in both tetanus and strychnine poisoning? Yes, because the action is same at the level of neuron. So that clinical features are seen in both of them. Then what is the feature that is differentiating them? Right? We need to know that. The first feature is muscle involvement. See, the first feature is muscle involvement. In strychnine poisoning, there is complete muscle involvement, while in tetanus, there is sequential muscle involvement. Okay. Why is this so? Because the tetanus poisoning, it binds to the nerves irreversibly. It cannot be unbound. So once there is contraction, there is no relaxation of that muscle. So sorry, I was explaining relaxation between convulsions. Let's start with this point. See, let's say at t is equals to 0 there is a convulsion, then after some time there will be relaxation, then again convulsion. This is a feature of what? This is a feature of strychnine poisoning, that there is complete relaxation between convulsions. This does not happen in case of tetanus. Why? Because the tetanus toxin which the bacteria releases, it binds irreversibly to these nerves. It cannot be unbound. While this strychnine can get accumulated at the spinal cord, and it also dissolves when its concentration decreases. So we have complete relaxation between two convulsions, while in tetanus there is incomplete. There is no relaxation, right? Second is muscle involvement. As I told you, in tetanus there is retrograde axonal transport. Means, let's say the bacteria has entered here, now it will go through the neuron to the spinal cord. Now every nerve is not the same in length and properties, so that's why every muscle will not be affected at once. There will be sequential involvement. While in strychnine poisoning, the poison is ingested via GIT and it reaches the blood circulation, and the poison is equally distributed here. So there is complete muscle involvement. Every muscle is involved. While in tetanus, based on the nerve which is controlling that muscle, and the nerves are of different lengths, so the poison is traveling like this via the nerves. So there is sequential muscle involvement. Now if you do not understand all this, you can just learn that muscle involvement is complete in strychnine while sequential in tetanus, and relaxation is not seen or incomplete in tetanus while there is complete relaxation in strychnine poisoning. Done. Now, what are the clinical features of strychnine? First is seizures. Why does the seizure occur? Due to that excessive muscle movement, there is seizure, and they are conscious because they are working at the level of spinal cord, not growing to the brain. As I told you, toxin does not cross the blood-brain barrier. Patient remains conscious. Now, see, what is this concept? I will make you understand. See, let's say right now the patient has an episode of convulsion. After some time, there will be relaxation phase. After that, again convulsion will start. But the previous episode was, let's say, 10 minutes. Then this will be 20 minutes, then the relaxation phase time will be reduced. The third episode, convulsions are of a longer time and relaxation phase is very short. So duration of convulsion increase but the relaxation phase decrease with each episode. Done. And death can also occur due to continuous muscle convulsions. Now, This we already studied the facial muscle spasm. The elevated eyebrows here you can see and the protruding eyeballs. This is rhesus sardonicus. We also see this type of posture. Basically everything is due to the muscle contraction which is not inhibited by glycine. So there is no relaxation, right? Posture scenes are opisthotonous, emprosthotonous and pleurostotonous, which is hyperextension of spine, then forward bending and lateral bending. What is the treatment? Treatment are to control convulsions. We give barbiturates, benzodiazepines and general anesthesia. Now here gastric lavage is not done. Why? See, if you do gastric lavage to remove this poison out, then you are irritating the muscles over here, and the muscles are highly irritated. The muscles are highly contracted state in here. You cannot disturb this muscle. So this was just a simple reason, right? The detailed reasons I am not covering here. It will take a long of time. So you can think like this, that muscles are already in a excited state. You cannot provoke them more. So don't do gastric lavage in the strychnine poisoning. Now, strychnine poisoning autopsy findings. We see an early rigor mortis. See, the concept is rigor mortis time is inversely proportional to the ATP. If you are having it after the death, the cells are having less ATP, then the rigor mortis will start earlier. So here, before death, there was excessive muscle contraction, right? So that was depleting the ATP. So after that, there are less ATP. Less ATP means rigor mortis will start earlier, right? Second one is post-mortem caloricity. Due to this excessive muscle movement or convulsions, there is heat generation which appears in the form of post-mortem caloricity. After the death, there is heat generation. Third is delayed putrefaction. See, in this their muscles are contracted and stiffened, so it is tough to decay that muscle. This was the simple reason why there is delayed putrefaction in strychnine poisoning.